With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Question has given us an assertion and a reason, and we need to tell if the reason, here is the reason, if the reason it is correct and the assertion is correct, and the reason given for this assertion is correct or not. The assertion here says that split gene arrangement complicates the definition of a gene in terms of DNA segment. So what are these split genes? Split genes, to understand this, we need to understand what are exons and what are introns. Exons, they are the part of any DNA or any mRNA which are going to expressed. So what happens is these exons, these are the part of any mRNA or DNA which encode for proteins. But these introns, they are silence genes, and these genes, they do not encode for any protein. And what happens is at the time of translation, when the mRNA is processed, these introns, these are spliced. Only the mature mRNA contains only exons. Now, in a gene, in DNA, only some part is exon, then this part will be intron, then some part will be exon, then this part will be intron. These are our exons, these are our introns, okay? This is how exon and introns are arranged in any gene, and that's why it is said that split gene arrangement, this type of arrangement is known as split gene arrangement. So this split gene arrangement, it complicates the definition of a gene because we need to, first of all, the DNA, it will convert itself into the RNA. RNA will undergo a splicing process, and from splicing process a mature mRNA will be produced. But what happens in bacterial cells, they do not have this type of mRNA, only exons are present on their DNA. So yes, this split gene arrangement, it complicates the definition of a gene. This assertion, it is correct. The reason given for it says that in eukaryotes, the monocistronic structural genes, this monocistronic, what is the meaning of monocistronic? Mono, as the name suggests, one, and cistron means gene. So monocistronic mRNAs are those mRNAs which encode for only single protein, and these are present only in eukaryotes. Whereas in bacteria, as I just told you, polycistronic genes are present, and these genes, they encode for many proteins. A single gene, it encodes for many proteins because it do not contain any introns, okay? So the reason says that in eukaryotes, monocistronic structural genes have interrupted coding sequence. Yes, because monocistronic genes, these are created after many processes. The RNA that is created after DNA, it undergoes splicing, then after splicing and mature RNA, it is created. So yes, it's a lot of work. So in eukaryotes, these genes, they have interrupted coding sequence, but also the presence of introns, they help in stabilizing our mRNA, whereas the RNA in bacteria, it is quite unstable, and that type of RNA, it is known as nascent RNA. So although in eukaryotes, monocistronic genes, they interrupt coding sequence. This is our coding sequence exon because they code for protein. This is non-coding sequence. So yes, they are interrupted, but it helps the gene to be stable, to be more stable. So although our reason, it is correct, but it is not a proper explanation of why gene split arrangement, it is there to stabilize our DNA. This is the reason why they are present there. So the answer to this question will be both assertion and reason are true, but reason is not a correct explanation of assertion.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.